up. For all of us, we ask that you use our ASCO affiliations for purposes of attribution in our meeting coverage. Our first presentation will detail the findings on the use of adjuvant chemotherapy guided by circulating tumor DNA analysis in stage 2 colon cancer. The lead study author, Dr. Jean T. of the Peter MacCallum Cancer Center in Melbourne and the Walter and Eliza Hall Institute, is presenting these findings, actually, I believe, as we speak, in the Clinical Science Symposium taking place just down the hall. So she provided a pre-recorded presentation for this press briefing. If you missed Dr. T in the press room yesterday and would like to talk with her in more detail about the findings, please reach out to a member of the media team in the press room to arrange a video. So we will start her presentation.
Hi all, I'm Jeannie T, a medical oncologist from Peter MacCallum Cancer Centre and Walter Eliza Hall Institute, Melbourne, Australia. The DYNAMIC trial is a study aimed to investigate how a novel blood test called circulating tumor DNA can be used to direct post-surgical therapy in stage 2 colon cancer. For oncologists, how best to manage patients with stage 2 colon cancer after surgery remains a clinical dilemma, because we know that surgery alone can cure over 80% of these patients. Chemotherapy is currently offered to stage 2 colon cancers with high-risk features, despite a majority not needing or benefiting from it. Therefore, a more accurate way to predict for cancer relapse has the advantages of limiting treatment to those who actually need the treatment, while allowing those who do not need treatment to be spared unnecessary chemotherapy. The goal of chemotherapy when given after colon cancer surgery is to eradicate any micrometastases if it's present. But the challenge is standard CT scan cannot detect these minuscule cancer deposits. The circulating tumor DNA or ctDNA blood test, which detects DNA released from cancer cells, can identify patients who still have microscopic cancer remaining after surgery, and thus a very high chance of cancer relapse if left untreated. The DYNAMIC study was designed to show that using ctDNA blood test to direct post-surgical therapy in stage 2 colon cancer can reduce the use of chemotherapy without impacting relapse risk. 455 patients were allocated to either ctDNA-guided management, where only those with the positive ctDNA blood result received chemotherapy, while patients with the negative results were observed, or to standard management, where treating clinician decides on the use of chemotherapy based on standard pathological criteria. The main findings were that the strategy of using ctDNA result to inform treatment almost halved the number of patients who received chemotherapy post-surgery from 28% down to 15%. And despite the lower proportion of patients receiving chemotherapy with ctDNA guidance, the likelihood of being alive and cancer-free at 3 years is the same as standard management, which is 92%. We also found that for patients with positive ctDNA results, the chance of being alive and cancer-free is 86.4% after treatment with chemotherapy. For patients with ctDNA negative result, the chance of being alive and cancer-free is 92.5% without chemotherapy. So in summary, in stage 2 colon cancer patients, ctDNA assessment after surgery allows a more precise prediction of relapse and patient selection for post-surgical therapy. The ctDNA-guided approach can reduce the number of patients treated with chemotherapy without compromising on their relapse risk. And given the favorable outcome in ctDNA-positive patients treated with chemotherapy, this well-defined high-risk subgroup of patients will likely derive substantial benefit from treatment. And finally, patients with a negative ctDNA result has a very low risk of relapse despite not receiving chemotherapy, suggesting that post-surgery therapy is unlikely to benefit this group of patients. Thank you.
Our ASCO content expert on this study, Dr. Kathy Eng, who is a gastrointestinal cancer specialist, provided some comments in the press release that you should have received this morning, and she will be available to answer questions about Dr. T's study following today's briefing. If you'd like to speak with her or Dr. T, please reach out to the ASCO press room. And from my perspective, I see this study as an important kind of new concept in cancers where, for the most part, we have really very good survival and outcomes. So early stage colon cancer, early stage breast cancer, where we've been able to achieve, in the majority of patients anyway, excellent outcomes. And now we're starting to look at ways that we can de-escalate therapy in a subgroup who we know are going to do well, while continuing the more intensive therapy or even escalating therapy in the group who we know are not going to do well with our conventional therapies. So this de-escalation concept of, we were able to show in the dynamic study that we could give less chemo, but using liquid biopsies, circulating tumor DNA, we could pick out a group that didn't need or benefit from the chemo and still have the same excellent outcomes. Now, the other end of it is those who had the positive circulating tumor DNA seen, so evidence of micrometastatic disease, we still need to do better. So excuse me, I do believe the results are going to help us guide our selection of who benefits from chemo and who can avoid it and all the toxicities of it in stage 2 colorectal cancer. So our next presentation highlights an analysis of sacituzumab